In
this talk, I'd like to make use of the fact that researchers in the early 20th century loved to use the image of Archimedes and the lever to talk about goals and resources needed to achieve them. Fritz Lacoeur, for example, underlined the importance of physiological test objects for chemists in search of hormones. Give me a test object, and I can optimistically begin the chemical processing of a hormone. In other words, chemists cannot start to pursue the goal of isolating a certain type of chemical entity without a certain type of biological resource. 13 years earlier, plant physiologist Friedrich Čapek said, on a more general note and from the perspective of someone interested in solving physiological problems, it is well known how important and how difficult it is in physiology to select the right point of the complex of questions to be investigated, in which the lever of the physical method is to be used first. According to Čapek, it was one thing to agree that the methods and concepts of physics and chemistry would or could potentially be helpful to solve biological problems, but it's an entirely different thing to match physical-chemical tools and insights with physiological questions. And according to British physiologist William Maddox Bayliss, this was not primarily a problem of specialist training. He observed that even students who have attended good courses in the physical sciences do not readily apply knowledge, the knowledge they're gained to physiological problems. And Frederick Gallup Hopkins says that chemists, on the other hand, have no aptitude for biological problems and do not recognize them. In what follows, I'm especially interested in this part of the picture. The place to put a lever, the special part in a bigger biological complex of problems, as Čapek suggested, and the thing that makes it possible to move the world, aka to analyze the subcellular process that is otherwise inaccessible. So this interest stems from an observation I made while studying researchers who in the 1920s and 30s combined resources traditionally associated with the biological sciences with resources associated with the physical sciences in order to study subcellular processes. And the observation is that historically it has been easier in each case of these four cases to identify potential levers and potential problem complexes, but way harder to find a specific experimental setup within which to mobilize the resource or resources developed in the physical sciences in order to gain access to the biochemical or biophysical process. And I can give you a short insight into these cases. And one important message or finding was that all these research projects were based on the assumption that a biochemical or a biophysical process underlies the physiological phenomenon of interest, and that therefore the tools of physical or organic chemistry could be helpful to elucidate these phenomena. Zoologist Selig Hecht, on the left side, for example, hypothesized that light might trigger a photochemical reaction in the retina, and that the products of this reaction somehow stimulate the nerves. And so he mobilized resources from the part of physical chemistry that is concerned with these kinds of chemical reactions, photochemistry. In the next case, groups of organic chemists and botanists in Utrecht and in Pasadena teamed up to identify the chemical substance responsible for plant growth. Here we have the hormones. And then in the United Kingdom, biochemist Rose Scott Moncrief collaborated with both organic chemists and geneticists in order to learn more about how genes control the synthesis of a particular group of pigments. And finally, in Prague, biologists and physicists joined forces to develop a method for biologists to determine the electric charge of biocolloids in the belief that knowledge of this property would help them to understand a variety of biological phenomena. And then looking at these assumptions in historical perspective showed that none of these assumptions were new for the time when they motivated the research projects I looked at. So for example, the idea that light could trigger a chemical reaction in the eye was around since the early days of photography, and it gained traction in 1877 when Fritz Boll found a chemical substance in the retina, in the cones of the retina of frogs, that bleaches out when exposed to light. So that could be a photosensitive substance. Then the idea that growth is somehow induced by certain chemical substance was already, this idea was already postulated by E.U. Sachs in the 1880s. And then 2 decades later, William Bateson's group studied the inheritance of flower color in a number of different plants. And although geneticists at the time tended to black box the question of how exactly Mendelian factors were involved in the production of their products, they assumed that genes somehow controlled biosynthetic pathways. And Muriel Wendell-Onslow, one of the collaborators of Bateson, suggested that one group of pigments was particularly interesting in this context, the anthocyanins, the pigments that are responsible for every coloration that goes from red or violet to blue in plant tissue. And she argued by combining the methods of chemistry and genetics, one could simultaneously gain insight in how genes act in the cell and how the anthocyanins are produced or synthesized in the plant. Wendell had studied the inheritance of flower color in snapdragons and observed that crosses of plants with white and ivory petals then give rise to offspring with crimson petals in whose sap we obviously find these anthocyanin pigments. And so she argued that for everyone that is interested in how this kind of pigment is synthesized, it's really helpful information to you to know what kind of input is needed to get at a pigment. And she thought that this case suggested that the pigment is formed from 2 colorless substances. There was no similar evidence for the last interfield assumption, but the work of Walter Nernst and others had shown how important electric charge was for physical processes. And many physiologists therefore consider it highly probable that this property also plays an important role in biological functions. So all in all, these, let me go back. These assumptions were widespread and somehow plausible, but nothing happened. So decades went by, and it was not possible to present empirical evidence to get from the assumption that there could be a relation between these events to a somewhat more plausible mechanism scheme. Until it suddenly was. So at least in 3 of the 4 cases, people found a way to do some experiments and to gain over these experiments and data that would help to support mechanism schemes. So for the rest of the presentation, I will therefore take a look at the exact resources and the capabilities that have made this step possible. So that was a long introduction. And these are the heroes of the story. And I want to concentrate on them, so don't be bothered by all the names I mentioned, because today it's about them. It's sea squirts, Siona intestinalis and clam, Mia arenaria, planktonic crustacean, Daphnia magna, and then different species of plants like Antherinum maius that produce these petals with the given color scheme. And oat, Avena sativa, but in a special stadium of its development, namely the coleoptile, which is the first protective sheath before, and it's the organism that is needed here in these kinds of research is the state in which the first primary green leaf doesn't come through yet. So just this part of the plant. So, and what I propose is that the special resource, the place where the lever is applied, is the phenomenon, and that means the behavior of the organism, and especially the behavior of the organism seen as a direct manifestation of the subcellular process going on inside. So let me just go through these animals and start with the oat coleoptile. Because in this example, we see a straightforward illustration of what I just said with behavior. According to Lacroix, a famous hormone researcher, the search for a hormone typically begins with a hypothesis developed in physiology, according to which a certain physiological activity is due to a specific organ and a yet unknown substance produced in that organ. Now, to find the substance, chemists need test objects that lack this organ and therefore do not exhibit the activity. Because then chemists could apply test substances and then decide, depending on the reaction of the animal, whether in their sample, so it's a retrospective inference of whether the substance has been in the test or not. And Fitzkogel, one of our actors in the case study and a friend of Fritzaker, said, not only need I a test object, I also need raw material that shows some activity, and then I can go on and enrich the product from there. So, and within 1 year, Kögel in fact achieved his goal. So when he said all this, that was his inaugural lecture, when he just came new to Utrecht, and he didn't start to collaborate with botanists yet, but it was very clear from the inaugural lecture that what he wanted to do is to find a hormone. And it was not really important what kind of hormone, in which system, whether in a plant or an animal, what physiological activity, but it had to be a hormone. And he succeeded. In the end of 1931, he claimed to have isolated the first known plant hormone, auxin. And that all was possible because he realized that the Utrecht botanists, without knowing it, had developed a suitable physiological test. It has long been assumed that the tip of the oat coleoptile produces a substance that induces growth, because when you cut that tip, the plant stops growing. Fritz Wendt, one of the PhD students in botany, then found that if you take this cut up tip and put it on a slice of agar, and then take one of the agar blocks and put it on one side of a decapitated coleoptile, that part of the plant resumes growth. And because the other part, the other side, doesn't have the substance and doesn't continue to grow, there is a resulting curvature. And now importantly, Wendt varied the amount of tips he put on the agar and could show that the curvature measured is directly proportional to the amount of hormone applied to the decapitated oat seedling. And this discovery is what made the coleoptiles a valuable test object for the chemists. They only needed to mix their test substance with the agar and keep away the tips, and then see whether the substance is able to induce growth in that test. And it wasn't the chemists, but the botanists who performed the tests, of course. They then could decide whether the fractions given to them by the chemists were active in the test. And by removing all the inactive fractions step by step, they finally succeeded in enriching the hormone until they got the purified crystal. So Kögel was celebrated by his peers. He received the Scheele Medal, the Emil Fischer Gedenkmünze. He got also nominated a couple of times by his peers for the Nobel Prize, never got it. Fritz Wendt, who invented the test, also got nominated for physiology though. And Kenneth Thiemann, who worked on the same project in parallel in Pasadena, together with a botanist trained in Utrecht, Hermann Dolk, and he got the Stephen Hales Prize in America. And through my research for another case study, I found this nice unofficial record of the euphoria this research caused among physiologists. Young George Wald wrote to his supervisor, Selig Hecht, in a letter from Berlin, "Have you been following the small quantities of high activity substances movement? I think this recent crystallization, analysis, and formalization of the growth substance, follicular hormone, testicular hormone with the work of vitamin A and other vitamins, is part of one of the most exciting developments that has ever come up in any science. From a humanistic standpoint, the mathematical mystical model that physics is plunging into doesn't stand a chance like yours." And also the ones involved in this kind of research were very happy in how it unfolded. Fritz Wendt and Kenneth Thiemann wrote in 1937, "Few fields can have benefited so much from the close interaction between the biological and the chemical approaches. And the remarkable discoveries on the chemical side have made possible equally remarkable progress in physiology." And that, to take you away from the main argument for a moment, is really not, or does not fit with a widespread historiographical narrative, according to which biochemical research in the early 20th century was characterized by the fact that biology had problems and chemistry had solutions, and that therefore chemists could colonize biology and just take all their good problems and solve them because they alone had the instruments to solve them. And in my dissertation, I argued that was more the overall argument, that it does take more than just chemical methods to solve the biological problems. It also takes biological methods. And on the other hand, biology offered resources for solving chemical problems. So chemists entering collaborations with biologists does not need to mean that they just came to solve biological problems. Maybe they came to solve chemical problems. And because they're both dependent on each other's methods and skills, they cooperated on equal terms, rather than there being a hierarchical situation. So back to the resources of biology. We've already seen that the key to the success of the research of plant growth was this finding by Fritz Wendt, the fact that growth correlates with the amount of hormone added to the decapitated coleoptils. This correlation, however, became only apparent because the coleoptils were grown in climate-controlled rooms, because Wendt used pure lines of the same harvest of one variety of Siegeshaber produced by Salöv, and because the data points represent the averages of 8 to 10 plants, because individual variety was still very high. And it's easy to see that Wendt followed some rules that his father, Professor and the leader of the botanical laboratory in Utrecht, had written down in textbooks. For example, he made clear that the goal of physiology and physiological investigations was to uncover the causal chain of events underlying physiological phenomena, and he joked that examining the influence of any factor on a life process is relatively simple. One needs only to keep all factors constant and then change one and wait for the result. And then he quickly admitted that it's not easy to keep the biological material constant. But what we can do is to work with pure material and to keep the conditions constant. And for growth, that means keeping temperature, humidity, and light constant. So, and this infrastructure was already in place when Wendt, the young one, began his dissertation. But in Pasadena, where Dog was brought by Thomas Hunt Morgan to be the general physiologist in this new group of biologists, the first thing he did when he arrived is to order these oat seedlings from Swallow and to buy the instrument needed to make a temperature-controlled room. So it's really clear that that was a really important part of the setup. And there in Pasadena, as in Utrecht, Dolk and his collaborator Thiemann used the correlation between amount of hormone and growth to identify the growth substance, and then also to use that substance, the purified substance, to make physiological experiments and then uncover the events that lead up to the cell wall being softened and then more water taken up, and so the cell increases in volume and thus growth happens. And another more abstract part of that project was to dig deeper into the structure activity relationship. So they took the plant growth hormone and its structure and then tried other substances with similar structures and tested for their activity and wanted to find out what is it exactly that makes these substances act. So now let us sum up what we have found so far. And I promise the next couple of case studies will be quicker. In a plant hormone case, everything depended on having a plant that grew consistently, meaning that grew in the same way under the same conditions, and that grew more if more growth substance was applied. So there was a hypothesis of what more hormone activity would mean to the physiological activity. So that correlation was safe, and it was really important to have a system where you could get rid of that hormone. So to introduce it new and then to test its action. And for that to be possible, and we can conceive of the original Avena test as a difference test in one plant, so to speak. So we have on the one side of the decapitated coleoptile, we have the factor, and on the other side not. And the difference in the growth results or brings about the curvature. So, and one of the properties of this Avena seedling that made this possible is the fact that this growth producing part of the organism is so strictly localized, and that it's in another part of the part than the part where it's acting. So it was easy to get rid of it and still have a functioning plant and see what happens if we add other substances. And the fact, and that was known from morphological studies earlier, that there's no cell division anymore in this stadium of the development. So cell division has already stopped, and all growth that happens is just cell elongation. So they knew that's just one activity that they're measuring. Und so das hat ein Bébé Borytropism LLE, the baby collector, is just came into existence in order for us to have this object to study tropisms and growth phenomena. And that brings us to the next couple of model organisms, in which, for which the same has been said. The main investigator here, Silly Hecht, wrote to his friend William Crozier that these creatures were specially created for being investigated. And now many of the things we can see are in parallel to what we saw for the growth hormone case. One thing is that these animals show a certain reaction to a given stimulus. And here the important stimulus is light, because we're dealing with light perception. And these animals show a reaction, they retract their siphons in a reaction to a sudden illumination. So a clear-cut and vigorous response on the part of the animal, but with a certain, so it takes a couple of seconds for them to react. So you can induce a stimulus and then take some time, then they retract the siphons. And Hecht argued that, so that was trivial to assume that what happened during that, between the induction of the stimulus and the reaction, somehow the reaction is a contraction of muscle because the nerves have been stimulated, and the nerves have been stimulated because the animal perceived light. So these are the processes that need to go on within the animal. And now Hecht knew from other experiments with other stimuli that these two processes are really quick. So for example, if you touch one of these animals, they also retract the siphons, and then they don't have this longer period to react. So he could assume that the time he measured for the whole reaction is basically the time that it takes for whatever happens in the sensory cell to happen, to convert the light stimulus into a nerve stimulus. And that was really exciting, because what photochemistry did at the time is to come out with laws of how certain factors influence the velocity of these photochemical reactions. Factors like light intensity, duration of exposure time, temperature, or wavelength of the light. And that has been a problem with other attempts to use this kind of knowledge for vision, because normally, for example, in vertebrate eyes, you have in the sensory organ rods and cones and different structures, so it's really hard to segregate light perception from color perception. But for these animals, that was easy, because they only perceived light and not colors. Hecht explained the significance of MIA for the study of the visual process lies in the fact that its photosensitive system is a simplified version of the eye. It possesses neither lens nor iris, and apparently only one kind of photosensitive cell. So we can be pretty sure to, again, measure just one kind of activity. Also, the fact that it's so slow is super handy. And so Hecht, inspired by what photochemists do, took these animals into a dark room and induced and applied light to them, but changed temperature, for example, or light intensity or wavelength of light, all these factors, and measured how long it takes them to react. And similar to the thing we saw for Fritz Wendt and the correlation between amount of hormone and growth, we have here an expectation from photochemistry that for a photochemical reaction, that is like the conversion of a photochemical substance into its precursors, for these kinds of processes, there's a law, the law of Bunsen and Roscoe, and it says that there's a constant amount of energy needed to elicit a photochemical effect. And energy is the product of the light intensity and the exposure time. So Hecht measured whether that is the case for his animals, and he could show that more or less that product is the same for different intensities of light and different reaction. So the reaction time gets longer with lower intensities. And really elegantly, by making these experiments to show that, he also could fulfill all the criteria of adequacy that in physiology were held for sensory studies. So he himself wrote this. He said, an analysis of sensory stimulation in order to be objective must take its data from the relations between the properties of the stimulating agent and those of the responses of the animal. And that was the term objective tells us or gives us a hint that this is the nomenclature, the objective nomenclature of sensory processes by Priabete and von Uexküll, was that we can't talk about the sensations of the animals. It's something that is not accessible to us, but we can talk about stimulus and response. And this is what we should do. And another of Hecht's peers, Leonard Rowland, wrote, if any hypothetical account of the mechanism of visual response is to be seriously considered, it must employ the definite structural and quantitative concepts of theoretical physics and chemistry, and the dynamical interactions which are involved must lead to the results which explain the phenomenon. And this too is something that Hecht does with direct experience, so he accounts for dynamical phenomena. And then just 2 more slides for Hecht. He argued that he can decompose this process of light perception into 2 sub-processes. He argued the mechanism of photoreception is not a single process. There is a fundamental division between the underlying machinery into an initial photochemical reaction and a consequent ordinary chemical reaction. Because he observed that in order for the animal to react in the end, it's not necessary to have light all the time. So it's just for this phase that a certain amount of light energy is needed, but then for that phase, it's okay to have the animals in the dark again, and still they would attract the siphons in the end. So that gave him a hint that this could be just another phase, a latent phase, as he called it. And then he just used everything that was known in physical chemistry about chemical reactions of this kind to argue that these are the reactions that underlie the 2 phases. For example, it was known that photochemical reactions don't get faster with the temperature going up. So he could show that. And in contrast to that, normal chemical reactions, they get 2 to 3 times faster with 10 degrees in temperature difference. And this latent period he could show is running by a factor of 2.5 times faster in a temperature that is 10 degrees higher. And made dynamical arguments and used the law of Arrhenius. And all that was applicable, and all that was the kind of data that he could use then to show that it was a plausible argument, a plausible schema that he presented here. So then back to the plants. Here we have chemists that are in the business of synthesizing anthocyanins, these pigments for the red to blue coloration of plants. And we have a biochemist, Rose Scott Moncrief, who what she did is to take living plants and then collect the petals, and from the plants and the cell sap, then she would try to isolate the pigments from there. So not synthesize them, but to take them out of the plant. And that wasn't easy at all. So that was one thing that people in Robinson's group were not able to do. And the geneticists with whom Scott Moncrief collaborated, they were interested in the outcomes of her work because they could then see what the material basis of the flower colors was. And the flower color was nothing less than the characteristic that they based their hypotheses on what kind of genotypes a plant has. So it was interesting for them to have like the real material basis of the characteristic than to just the appearance to the naked eye. For the chemists, on the other hand, it was interesting to collaborate with Scott Moncrief because they wanted to have a comparison between what they synthesized and what the plant produces. So they could show when the isolates in the synthesis have the same characteristics, that they managed to synthesize something that is a natural, it has the same characteristics as a natural pigment. So both sides were interested in collaborating with Scott Moncrief. Scott Moncrief, on her part, had more ambitious goals. She wanted to find out how individual genes influence the production of anthocyanins in the plant. And to do this, she combined the results of her work with the results of the geneticists and the chemists' work. So from the geneticists, she got hypotheses about the genotype of the plants, from which she isolated the pigments. And from the chemists, she got insights into the structure of these pigments. And what she found was spectacular. She could show that individual genes or genetic differences correlated with structural differences, and therefore could make the argument that the addition of this OH group, for example, is a result of the dominant factor A and its activity in the plant. And again, we have a good idea for the researchers that what they study is just one activity, namely the pigment production. And because of all the work of the geneticists have done in the 30 years before, they also knew they were able to intervene on the genotype of the offspring. So they could cross genotypes and then bring about plants that produce pigments at will. And they had some hypotheses about what the activity of the genes are, because genes are basically defined by their activity. So that is interesting to compare with the other two cases we had, because here the intervention as well as the expectation of what happens when we intervene on the system in a given way comes from the biological sphere and not from the chemical sphere. The chemical sphere is important to see what the result of the activity is, because the chemists were able to say, oh, this is that kind of pigment, and that pigment has this structure. So in this case, what made these good experimental organisms is just the fact that they produce pigments, and that for contingent reasons, more or less, the chemists and the geneticists were both working on these pigments. And the chemists had developed methods to characterize them and identify them, and to learn about their structure. And the geneticists knew about the genes responsible for the production of these pigments. So Scott Moncrief said the two advanced lines of research, so fortuitously being undertaken at the same period, complemented each other and were the first systematic uncovering on an extensive scale of the biochemical nature of gene action, and thus set in motion the contribution which biochemistry was to play in genetical research from then onwards. And John Burton Sanderson Dane, who was the supervisor and the leader of the research of the John Innes horticultural institution at the time explained in a later talk he gave at the Children's Center how important it was to have, besides the chemical methods, the other side. And I hope this is working. This is him talking.
It is, however, a postulate of physiological genetics that any difference, which is usually determined genetically, can also be determined by non-genetical causes. If that were not the case, genetics would be an inscrutable mystery. We could never know the causal path between a gene and a scorable character.
And now the last case study, which is really quick because here they didn't find such a correlation. Although they had Daphnia magna in the center of all their experiments, and they had some way to intervene by applying dye from which they know what kind of chemical charge they had, and they thought that then dyeing the animals with that stain would reveal the electric charge of the biocolloids, and that would eventually explain everything. They had no physiological activity that they could correlate what they think happened on the chemical side or on the physical chemical side. So they could intervene. Maybe I'll show the example first. That is the closest they got to such a correlation. They found out that the mortality of Daphnia magna correlates with, or there's two peaks of the mortality in a solution that in places where the solution has a low dielectric constant. But that in a way didn't get them anywhere because it was dyeing. What kind of activity is that? Maybe not one. It's detectable, but they had no hypothesis of how the dielectric constant of the solution being low led to the death of the Daphnias. So there basically was no mechanistic scheme that they could support with data. So it was hard to come up with the data to support a non-existing scheme. And from all these cases, how am I doing with time?
You can take another 10 minutes if you want, but it depends on how much discussion you want.
Yeah, good. So maybe I just wrap up and then say why it's interesting, and then we can. So in all the cases where it worked, I would argue it worked because people found a correlation between what they could observe on the outside, on the physiological level, and what they thought was going on the biochemical or biophysical level, something that they did not have the instrument to observe. And that correlation gave them something like they were allowed to use whatever they measured about the physiological activity to take to be a property of the biophysical or biochemical process. And that seems a bit trivial, but a couple of things that are maybe not trivial is that, so these are all the correlations that I identified, and they always happened very early in the research project. So in the first couple of weeks, Selig Hecht did his experiments in La Jolla on the beach. And even before Krüger started to cooperate with the botanists, and after 1 year, it was clear that there was this correlation. So it was always early into the research project that people knew maybe there's something to get at, and it gave them a hint that it would be worthwhile to invest in that line of research more. So I think that is interesting. Then the overall picture we get is that all that happens in an experiment is we have this biological, the experimental organism. We vary the conditions, we intervene, we detect on the level that we can detect what happens, and we have some idea of what happens on the lower level. And I'm happy to report that there is a contemporary physiologist who said something like that. He said plants and animals are the biologist's instruments, for they alone exhibit the phenomena which are the center of his interest. It is the initiation, correlation, and succession of the changes manifested by his instruments which he's endeavoring to understand. So I really like this quote because I think it underlines nicely what I tried to show. And then there's other quotes that go in this direction. For example, Frank Libby said the zoologist should know the wealth of animal life for its suggestiveness and for the opportunities of unique and specific animal forms for successful experiment. And then A.V. Hill said to find suitable organisms, physiology should be in close contact with zoology. We might add with botany, simply from the point of view of expediency, of finding more suitable animals and cells and functions for story. I think these quotes show how really difficult it was to get organisms that show the activity that we want. And then Rudolf Höber said, yes, it's unlike in chemistry or in physics, we cannot just produce from scratch the experiment we want. And this is why the physiologist often scours the entire living kingdom of nature to see if nature, out of all her manifold forms, doesn't throw and one like throws the physiologist into his lap a form in which she herself, nature, has already demonstrated desired, otherwise impractical experiment. And then he said, such discoveries delight the physiologist. They lure him across national borders and let him across oceans to foreign places of research with an eagerness that seems strange to the distant observer. And judging from the cases I looked at and the organisms used there, I can say that people didn't really scour the entire living kingdom of nature to find a certain form. It was more that they already worked with that form and then realized that they were thrown in their lap a form that was really useful to investigate a certain phenomenon. So Silicact already wrote his PhD about another sea squirt. The laboratory in Utrecht worked with Amina coleopters for 20 years by that time, and that was their experimental organisms for all kinds of researchers. And then the whole story with the anthocyanins all just was possible because the Bateson research program worked with these kinds of plants for a long time and looked especially for these kinds of characteristics. And also Josef Gickelhahn, the biologist in the Prague case, already worked with Daphnia before he decided to take Daphnia as the animal to stay in his experiments. So I would say, so that's one thing to say. They didn't actively search because maybe that's pointless. It's so hard to find it. The more realistic way is to just realize that you have that form that lends itself to the investigation of a certain phenomenon. And then I would say such discoveries delight the physiologist and the chemist, and this is mostly seen with Kögel, the chemist in Utrecht, who was so delighted by this Avena test that he readily decided to invest all he had basically with his group to look for that hormone. So that is interesting about these kinds of experimental organisms. They inform the choice of research program. They inform decisions to collaborate with certain experts. And they can be a reason to change methodological norms, and maybe this is what I'll end with. because in the case of plant hormones, it was super clear to the botanists that if we want to find this growth substance that we don't know yet, we need to look for a substance that induces growth in decapitated coleoptiles, but also one that is likely to be produced in the plant. And Köhler got rid of that second requirement. He said we can use whatever shows some activity. So he tried all kinds of substances and found out that human urine is especially potent in the oat test. And then he took human urine as the raw material from which he then in the end isolated the auxin, and that would be something that the botanists never would have thought of, but they were okay with because it worked. And then there's another example of the other chemist, Robin Robinson, who in the first place was really not happy with the idea of collaborating with geneticists. He said what geneticists try to do with genetical methods, find some insights into synthetic pathways is like cracking a walnut with a sledgehammer. And then really 3 years later, oh no, that's another, well, he changed his mind really quickly and then retrospectively said that geneticists can do chemical experiments in vivo. By inducing a new gene, for example, a methylation factor, he can observe its operation on a new anthocyanin. This science, meaning chemical genetics, is only in its infancy and is probably from research. It is probably from researchers in this field that we shall get the fullest knowledge of the chemical processes that modify the anthocyanin precursors or the anthocyanins themselves. And the situation I alluded to earlier when I said within a really a short period of time, he changed his mind because in 1933, that is 4 years after he said the thing with the sledgehammer, he went to the International Congress of Applied Chemistry, and he said to his peers, we cannot have, so from now on, we need biochemical insights and data to support hypotheses about synthetic pathways. The details of the biogenesis of plant products will only emerge as a result of specific biochemical investigations and from what we already know of the intimate mechanism operative in the laboratory of the living cell. Analogies from the chemistry of the test tube may prove highly misleading. And that was really new by the time. It was believed that if you could synthesize a natural substance in vitro and it has the same characteristics, then you probably found the way that the plant or the animal is using as well. And Robinson said, no, not at all. And I would say he said that also with a view to Paul Kara, who sits here, who also worked on anthocyanins and who didn't collaborate with geneticists and didn't have that kind of collaboration with access to biochemical data. So to conclude, the place to put a lever are these moments where we have a correlation between something we can observe on the physiological level and something we think goes on the biochemical or biophysical level. And we have seen that finding such a place where to put a lever also can change people's idea of what resources are needed to achieve a goal according to the norms. So with that, I'm finished.